Have you ever heard of Kaposi Sarcoma-associated herpes-virus or KSHV? This virus, also known as human herpes-virus 8, is a member of the gamma herpes-virus family and is associated with several types of malignancies. It's the etiological agent behind Kaposi sarcoma, a type of vascular tumor, as well as two B-cell lymphoproliferative disorders, primary effusion lymphoma and multicentric Castleman's disease. KSHV can be transmitted in a variety of ways, including through saliva, sexual contact, blood transfusions, and organ transplantation. Certain groups of people are more susceptible to KSHV-associated malignancies, particularly individuals who are immunocompromised, such as those with HIV/AIDS, recipients of solid organ transplants, or those undergoing immunosuppressive therapies. Now that we have a basic understanding of what KSHV is, let's delve a bit deeper into the molecular mechanisms involved in the diseases it causes. First, let's talk about how KSHV enters cells and establishes latent infection. The Kaposi sarcoma-associated herpes virus, or KSHV, employs a rather cunning strategy to invade our cells. It begins with a simple handshake, the virus binding to heparin sulfate, a component found on the surface of many cells. This initial interaction is like a secret handshake, allowing the virus to cozy up to the cell. But this is only the first step. KSHV also interacts with integrins, proteins that act as bridges for cell-cell interactions. These integrins essentially roll out the red carpet, further facilitating the entry of KSHV into the cell. The virus also takes advantage of other cellular receptors in its invasion strategy, showcasing its adaptability and cunning. Once inside, KSHV doesn't immediately cause havoc. Instead, it goes undercover. Like a secret agent, it establishes what we call a latent infection in various cell types. In this state, the virus is essentially sleeping or dormant. It's present in B cells, endothelial cells, and epithelial cells, but it's not causing any immediate damage. However, don't be fooled by this seemingly peaceful state. The virus is merely biding its time, waiting for the right moment to awaken from its slumber. When it does, it can trigger the development of diseases like Kaposi sarcoma and other malignancies. So once the virus enters the cells, it can lay dormant. But how does it cause disease? This is where KSHV's viral oncoproteins come into play. These viral saboteurs, encoded by the KSHV itself, have a knack for dysregulating cellular signaling pathways involved in cell proliferation, survival, and angiogenesis. Let's consider a few of these viral oncoproteins. One is LANA, which stands for latency-associated nuclear antigen. LANA plays a pivotal role in maintaining viral latency and manipulating cellular gene expression. Then we have V-cyclin, a viral version of cellular cyclins that regulates cell cycle progression. This oncoprotein can accelerate cell cycling, leading to uncontrolled cell division, a hallmark of cancer. Next in line is VFLIP, short for viral splice inhibitory protein, an important player in inhibiting apoptosis, which is the process of programmed cell death. By blocking apoptosis, VFLIP ensures the survival of infected cells, providing a safe haven for the virus to thrive. Finally, we have VGPCR, a viral G-protein coupled receptor. VGPCR can activate multiple cellular signaling pathways, including those that promote cell proliferation and angiogenesis, the formation of new blood vessels, which is critical for tumor growth. These viral oncoproteins target various pathways, including NF-κB, PI3K/ACT, MAPK, and JAK-STAT. By hijacking these pathways, they promote cell transformation and tumorigenesis, the process by which normal cells turn into cancer cells. These oncoproteins essentially hijack the cell's normal functions, leading to cell transformation and tumor formation. But how does the virus evade our immune system? KSHV has developed several strategies to evade our immune system. One such strategy involves manipulating antigen presentation. The virus can alter the function of major histocompatibility complex proteins, which are key players in the immune system's ability to recognize and respond to pathogens. By doing this, KSHV effectively becomes invisible to our immune defenses. Another clever trick KSHV uses is interfering with cytokine signaling. Cytokines are molecules that facilitate communication between immune cells. When KSHV disrupts this communication, it can weaken the immune response, allowing the virus to persist. Lastly, KSHV can downregulate host immune responses. This means it reduces the intensity of the immune response, further securing its survival within the host. This combination of evasion tactics not only allows KSHV to persist, but also contributes to the development of malignancies. With these immune evasion strategies, KSHV can persist in the body and contribute to the development of malignancies. KSHV doesn't stop there. It also promotes angiogenesis and transforms B cells. Now let's dive deeper into these processes. When KSHV infects endothelial cells, it induces the expression of pro-angiogenic factors like vascular endothelial growth factor or VEGF and interleukin-6 or IL-6. These factors stimulate the formation of new blood vessels, a process known as angiogenesis. This is a key step in the development of Kaposi Sarcoma, a highly vascular tumor. But the virus's influence doesn't end with endothelial cells. KSHV can also infect and transform B cells. With the help of viral proteins such as VL6 and VFLIP, the virus promotes B cell proliferation and survival. This transformation can lead to the development of two B cell lymphoproliferative disorders, primary effusion lymphoma and multicentric Castleman's disease. These processes highlight the virus's ability to manipulate our body's cells in quite profound ways. By promoting angiogenesis, KSHV ensures the tumors have an ample supply of nutrients, enabling their growth. And by transforming B cells, the virus is able to generate new sites for its replication, furthering its spread within the host. Unfortunately, there is currently no effective vaccine or specific antiviral treatment for KSHV infection. However, understanding these molecular mechanisms can help researchers develop new therapeutic strategies. These strategies could potentially disrupt the virus's life cycle and curb the development of KSHV-associated malignancies. In conclusion, KSHV is a complex virus that uses a variety of strategies to invade our cells, evade our immune system, and cause disease. It is a cunning adversary capable of establishing latent infections, dysregulating cellular pathways, and promoting angiogenesis. The virus is particularly dangerous for immunocompromised individuals, leading to malignancies like Kaposi Sarcoma, primary effusion lymphoma, and multicentric Castleman's disease. While there is still much to learn, understanding these mechanisms is a crucial step in the fight against KSHV and the diseases it causes.